Okay, I think we should get started. Hello all and welcome to the ZOOMinar. Today we have two outstanding speakers from Japan, Dr. Tomohide Saio from Tokushima University and Takaomi Saido from Riken, Wako, Japan. Here's a brief introduction to the first speaker, Tomohide Saio. Dr. Saio completed his bachelor's, master's, and PhD degrees from Hokkaido University. His research was carried out in Professor Inagaki's lab at Hokkaido University and postdoctoral research at Rutgers University. In 2014, he went back to join Hokkaido University as an assistant professor in the Division of Chemistry. He was recently promoted to the full rank of professorship in 2020 at Tokushima University. He is the recipient of Young Scientist Award at the 15th annual meeting of the Protein Science Society of Japan. His main focus is on the structure, dynamics, and function of molecular chaperones. With that brief introduction, Saio-san, floor is yours.
Thank you very much for kind introduction. I'd like to also thank the Professor Rams for kind invitation to this wonderful Zoom meeting, Zoom seminar. So today I will be talking about the regulation of protein folding and protein assembly by the regulators, the chaperones. And as many of you know, the chaperones recognize unfolded proteins and the protein folding intermediate to support the folding into the native fold and prevent the protein folding amyloid formation, and prevent the protein aggregation or amyloid formation. And on the other hand, the recent studies have unveiled a new functional aspect of the chaperones, and that is the regulation of the functional protein assembly in liquid-liquid phase separation. In both cases, the regulation is achieved through weak and dynamic interaction between the client protein and the regulators. These weak and dynamic features make it difficult to obtain the structural information for the complex by the X-ray crystallography or CRIEM. And the lack of the structural information about chaperone client complex has prevented the elucidation of the mechanism. Here in our study, to investigate how the molecular chaperones are working, our main research tool is NMR, the solution NMR, that is good at investigation of weak interaction of dynamic molecules at atomic resolution. So despite of those advantages, one of the major limitations in NMR structure study was size limitation. So this is a histogram showing the PDB deposition number and the protein sequence length. And as you see here, most of the protein structures determined by solution NMR is limited below 300 amino acid length or 30 kilodalton. And if you study the protein larger than 30 kilodalton by solution NMR, you often suffer from the low sensitivity and signal overlap, and you often end up with getting this kind of poor NMR spectrum. And with this poor quality NMR spectrum, it is difficult to obtain the high resolution structural information of the protein. On the other hand, we explored several NMR techniques for targeting large proteins, including methyl selective isotope labeling, in which all of the protons except methyl groups are deuterated, and only the methyl groups are NMR active in the protein. And with this labeling method, coupled with other techniques, including methyl torosy experiments, high resolution, high sensitivity spectra can be obtained even for large proteins. And in my study so far, several large proteins, mostly chaperones, have been investigated by NMR. So with these NMR techniques, I investigate interaction, structure, and dynamics and kinetics of chaperone client complex and try to unveil the mechanism at atomic resolution. And in my talk today, I have two topics about NMR study of the chaperones. And the first in protein folding, and second is in protein assembly. So let's move on to the first topic, chaperones in protein folding. So as I mentioned at the beginning, chaperones support protein folding and prevent misfolding and aggregation through the recognition of unfolded proteins or folding intermediate of the proteins. And in the cell, several molecular chaperones are working. And on the left-hand side, the major molecular chaperones in bacterial cytosol are shown. And here, the newly synthesized proteins emerging out of the ribosome is first recognized by this blue chaperone, TF, trigger factor. And after treatment by trigger factor, some proteins can succeed to form the native structure. But some other proteins may need further assistance by the other molecular chaperones, including DNAK or GLOEL. And if the protein has signal sequence, then it is transferred to the other chaperones of SEC-B and SEC-A for translocation. So here, the multiple different types of the molecular chaperones are working in the cell, forming a network. In this network, each of the molecular chaperones has very distinct function and distinct activities. And here I show you one of the examples of showing the distinct activities of the molecular chaperones. And in this experiment, in vitro MVP refolding is performed in the absence and the presence of the molecular chaperones. And here, as you see, in the presence of the trigger factor TF, the refolding speed and yield of the MVP was accelerated, showing the 4 days activity of trigger factor. On the other hand, in the presence of the SEC-B, the folding is significantly suppressed, showing the whole day's activity of SEC-B. So even though these two proteins are both classified as molecular chaperones, but the effect in the refolding experiment was quite different. So now my study is trying to understand the mechanism for distinct activities of these molecular chaperones. So first of all, we tried to investigate the binding sites of the molecular chaperones on the client protein. And in this experiment, the unfolded protein, client protein, the 4A, was N15 labeled, and the NMR spectra of the 4A fragments are measured in the absence and the presence of the trigger factor chaperone. And as you see, in the presence of the addition of the trigger factor, several specific resonances are perturbed. And in this experiment, each resonances corresponds to each residues in 4A. So by monitoring the peak intensity change by the addition of the trigger factor, we can monitor the binding residue by residue. And by taking the peak intensity ratio, the binding sites of the trigger factor on the 4A are identified. And the major 7 binding sites were identified. So if you look at the binding sites, which are highlighted in gray here on the hydrophobicity plot, and you will see that the most of the binding sites of the trigger factor chaperones are highly hydrophobic. And based on the same experiment, the binding sites of the SEC-B on 4A are also determined. And you see that although there is few difference, but still majority of the binding sites are common in both cases. And also the affinity estimated by the ITC experiment shows that these two chaperones recognizes the client protein in a similar affinity. So by just looking at the binding sites and affinity, we were not able to explain the difference of the activity of these two chaperones. So next, we proceeded to determine the structure of the chaperone client complex by NMR. And in this case, the structure of the trigger factor in complex with the client protein 4A was determined based on these noisy experiments. And I don't go into details today, but the important finding from this structure is that the client protein, the hydrophobic regions of the client proteins were recognized by the multiple different binding sites on the trigger factor chaperone. So based by these the multiple bindings, the client protein is kept unfolded and elongated, and that's how the molecular chaperones prevent the aggregation and misfolding of the client protein. So this structure suggests that in complex with the chaperones, the folding is not allowed and kept unfolded. On the other hand, N15 relaxation dispersion experiment showed that this complex is quite dynamic, and it undergoes fast on and off exchange. And the exchange rate Kx was estimated as 1300 to 1500 per second. So this data shows that the chaperone client complex is highly dynamic, and it undergoes binding and release exchange all the time. And also for the SECP chaperone, my former colleague, Dr. Huang Chendong, determined the structure of the SECP client complex by NMR. And this structure shows that the SECP tetramer has disc-shaped structure, and the client protein wraps around this SECP disc. And although these two chaperones, TF and the SECP, has common binding sites on the client protein 4A, but the complex structure looks quite different. And furthermore, another important difference between the two chaperones was seen in the binding kinetics. And the binding kinetics with the client protein was measured by BLI experiment. And we found that the SECP has a faster K-on and slower K-off compared to the trigger factor. And I think this difference in the binding kinetics can explain the difference in the activity. So as structure shows, the client protein was kept unfolded on the chaperones. However, the chaperone client complex is dynamic, and the client protein is sometimes released, and then it is allowed to proceed with its spontaneous folding. On the other hand, when the molecular chaperone has the faster K-on, and it quickly recaptures the client protein, and then the client protein has not enough time to proceed with its spontaneous folding, so that these are the chaperones with faster K-on has the stronger whole days activity. So if it is the case, then one can expect that the folding rate, spontaneous folding rate of the client protein itself can affect the 4 days whole days balance. And indeed, the experiment shows it is the case. And in this experiment, the MVP mutant that shows the slower folding rate compared to the wild type was used. And in the refolding of this MBP mutant, even trigger factor can act as holdase. So this data shows that if the protein folds slower, the same chaperone can act as the holdase. So our result shows that the chaperone activity is determined by the balance between the folding speed of the client protein and binding kinetics with the chaperones. So for example, when the chaperone has faster K-on, or when the client protein has slower folding speed, then the chaperone should exert a stronger holdase activity. So I call this the activity kinetics relationship. So the chaperone has very promiscuous sequence recognition of the client protein, but this diversity of the activity of the molecular chaperone should be exerted by this difference in the binding kinetics. And I'd like to show you another example of the activity kinetics relationship as seen in this trigger factor monomer dimer comparison. So trigger factor can form a dimer in solution. And based on the structure of the trigger factor dimer that we solved by NMR, and we designed the monomeric mutant that mostly exist in a monomer in solution. And we compared the binding kinetics with the client protein, and the data shows that the trigger factor dimer has faster K-on compared to the monomer. And as expected from this binding kinetics, and trigger factor dimer showed stronger holdase activity. So the data shows that even the same chaperone can modulate the activity by the modulation of the binding kinetics coupled with the oligomerization. So let's move on to the second topic, the chaperones responsible for the regulation of the functional protein assembly, liquid-liquid phase separation. These are called the LLPS chaperones. And LLPS or biomolecules, the proteins and nucleic acids, is driven by weak and transient interaction so that the specific molecules are assembled together and separated from the other molecules to form the membraneless organelles. And the membraneless organelles formed by LLPS make smaller compartments in the cytosol or in the nucleus. And here, the LLPS is often regulated by LLPS chaperone that maintain the balance between the assembly and the dissociation of the LLPS proteins, and accordingly prevent the protein aggregation and amyloidogenesis. And recent studies unveiled the several LLPS chaperones responsible for the regulation of the specific LLPS proteins. And here, our target in the study is the LLPS related to ALS. And as shown in this graph, ALS-related mutations are seen in multiple proteins. On the other hand, one key feature is that the majority of these ALS-related proteins have low complexity LC domain, consisting of 4 or 5 amino acid types among 20 types. And the LC domains are often responsible for LLPS droplet formation. On the other hand, the largest population in familial ALS is related to C9orf 72 repeat expansion that result in the generation of the dipeptide repeats, including proline arginine or glycine arginine repeats. And although the mechanism how these dipeptide repeats cause toxicity was unknown, the several recent studies suggested that this peptide involves in the LLPS regulation. And here I show you the scheme of the LLPS regulation and dysregulation of ALS-related proteins. LC proteins, including fuse, is thought to be in equilibrium between monodispersed and the assembled into LLPS droplet, and the balance is mediated by LLPS chaperone. In this case, a protein called capital-2 or transportin 1. And capital-2 was not actually a molecular chaperone and was originally identified as a protein responsible for nuclear transport. But the recent studies unveiled a new functional aspect as a LLPS chaperone. And capital-2 binds to fuse through tight recognition of nuclear localization signal, NLS, and weaker and multivalent recognition of other regions of fuse, and accordingly suppress the LLPS formation. And in our study, in collaboration with Dr. Mori and Dr. Yoshizawa, and we identified that PR repeat peptide inhibits the function of capital-2, and we investigated the mechanism how the PR repeat peptide inhibits the capital-2 function as LLPS chaperone. So first, I'd like to show you our data showing the inhibition of capital-2 function by PR repeat peptide. And in this experiment, the hydrogel of MCHERI labeled fused LC protein was prepared and incubated with GFP labeled fuse, so that the accumulation of the polymerization of the fuse was can be investigated by the GFP fluorescence microscope. So as shown here, in the presence of the capital-2, and the polymerization of the fuse is suppressed, but by the addition of the PR peptide, then the polymerization is retrieved. In the microscopic observation of the fuse droplet, the same trend was observed. So this data shows that the PR repeat peptide inhibits the capital-2 activity and dysregulates this chaperone function. And to see the direct binding between capital-2 and PR repeat peptide, and then we exploited SECMAS experiment, and in which the capital-2 was mixed with the MVP PR18 repeats, and the size of the complex was estimated by the light scattering after the size exclusion chromatography. And the data shows that the complex was estimated as 130 kilodalton, and that corresponds to the 1-to-1 complex. So this result shows that this PR repeat peptide directly binds to capital-2 at 1-to-1 ratio. And next, to investigate the binding site of PR repeat peptide on capital-2, we exploited NMR. And in this experiment, one signal corresponds to one methyl group of the capital-2. And as you see here, the several specific resonances of the capital-2 are perturbed by the addition of the PR peptide. So, and then next we wanted to assign these resonances of capital-2. However, due to the large size of the and the limited yield of the capital-2, it was difficult to obtain the methyl assignment at that time. So we decided to obtain the information through the comparison of the perturbation patterns with other known binding partners. So the first the reference is the M9N inhibitory peptide that is known to bind to this NLS binding site of the capital-2, in this cavity here. And as a second reference, we use this fused delta NLS that has weakened multiple bindings to the capital-2. So this slide shows a summary of NMR analysis with the reference spectrum. And I don't go into details today, but the key findings are that some resonances are perturbed only by PR20, but not by M9M, while some others were perturbed by both PR20 and M9M. So these observations indicate the binding site for PR20, PR repeat peptide, overlaps with that for M9M and NLS. So, okay. So this indicates that a PR repeat peptide partially occupies the binding site for the NLS. So this NMR observation is confirmed by ND simulations, showing that the PR peptide binds to the negatively charged cavity of the capital 2. So our data showed that the PR binds and occupies NLS binding site of the capital 2 and inhibit the binding of NLS. So this is a summary of our talks in the second topic. And the capital 2 is known to work as a chaperone and promotes the dissociation of the fuse from the droplet. And we found that the C9-derived PR repeat peptide binds to the capital 2 and partially occupies NLS binding site. And this interaction prevents the interaction with the fuse, so that the activity of the capital 2 was suppressed. So let me finish my talk with acknowledging the people who are involved in this study. So this is my current team in Tokushima University, and Dr. Kumashiro and Mr. Kawagoe are currently conducting research project of the trigger factor chaperone. And Ms. Kawamukai and Dr. Masaki and Ms. Kato are now conducting capital 2 projects. And then the trigger factor chaperone project was started in Rutgers University when I was working in as a postdoc in Babis Kautimus lab, and continued and developed in Japan. And I'd like to thank the people in Hokkaido University, my former position as an assistant professor, and especially Professor Ishimori and Dr. Kumeta for NMR experiments. And I'd like to also thank the collaborators in the other institute, including Dr. Okumura and Professor Muraoka, who gave a talk last week, and Professor Lee in KBSI. And I'd like to also thank the financial support from these organizations listed here. And finally, I'd like to thank you for your kind attention, and I'm happy to take any questions and comments.
Thank you very much, Dr. Saio-san. Please post your questions for the speaker in Q&A folder. They will be discussed. I have a question. So it's very nice talk, very interesting topics. You mentioned about the dimer, TF dimer has faster kinetics and stronger holding activity. Is it because just because the surface area, or how does it, what kind of, can you provide more insights into monomer versus dimer? Why is it more stronger and faster?
Yeah, thank you for the question. Actually, this is very important questions. And actually, we are also trying to understand, want to explain why this difference is generated. So one possibility is that in the dimer, the client binding site is assembled into the closed, assembled close together. So this can affect this can somehow promote the bindings. That was the one possibility. So, but actually, we have no evidence for this explanation. So we are still working on to explain this.
Do you expect the dimer to be in equilibrium with the monomer in your sample?
Yep. Thank you for the question. Actually, this is the KD of the dimerization is about 1 or 2 micromolar. So, and the like a cellular concentration of the trigger factor is estimated as 50 micromolar. So that means the trigger factor is mostly exists as a dimer in the cytosol. So, but actually this the trigger factor, when trigger factor binds to the other molecules like client protein or the ribosome, and the trigger factor is monomer. So there should be some equilibrium between monomer and dimer and different locations.
I don't see any questions. Let me ask you another question. And this initially you showed a 2D NMR spectra of this client protein 4A with the TF and the SEC-B here, right here. You show a complete shift in the resonances, but you only plotted the intensity. So the chemical shift changes seems to be dramatic on the leftmost one for the region 1 to 122. So can you comment on that?
Oh, you mean the chemical shift difference in this spectrum?
Yeah.
Yes, you see, yes. As you pointed out, some resonances show the chemical shift difference, but mostly it shows the signal intensity reduction. And this intensity reduction is probably due to the size increase upon the binding to the trigger factor, because in this experiment, the 4A is protonated. And this is not a Trozy experiment, but this is HSQC experiment. And of course, we have to consider about exchange broadening as well. Mm-hmm. And for the resonances with the chemical shift difference, maybe the binding kinetics is faster, and so maybe like this difference can highlight the difference in the binding kinetics as well.
Very interesting. Okay, there is a question in Q&A. You can also take a look at it. I'll read it for you. From Velayudham, it says, he says, great talk. What is the effect of temperature on TF dimer formation?
That's interesting. Thank you for the question. We haven't looked details about the temperature dependence. For this, I have no information about the temperature independence. But the interesting thing is that the thermal stability of the trigger factor molecules is not so high. And if you increase the temperature to like a 50 or 40, even 40 degree, and then trigger factor starts unfolding. So it looks that the trigger factor is very flexible and flexible and proteins. So yeah. So maybe, so if you look at the temperature dependence for the dimerization, it should be some dependence. So, but we haven't tested yet.
So another question from Velayudham is there any endogenous inhibitor for TF dimer?
Yes, actually, when trigger factor binds to the ribosome, then it monomerizes. Or when the trigger factor binds to the client protein, then the monomeric form is stabilized. So when something binds to trigger factor, then the dimer dissociates into monomer.
There's a question from Beena Krishnan. Trigger factor binds its clients when it is coming off the ribosome. while Seg-B binds its clients post-translationally. How do you see your data in the physiological context when thinking of a competition of a common substrate for the two chaperones?
Yeah, thank you very much. This is a very interesting, important question. Actually, yes, as you pointed out, the trigger factor binds to the client protein when the protein is coming out of the ribosome. But there's the previous studies, biochemical study showed that the trigger factor can depart from the ribosome with the client proteins. So it is waiting for the client protein coming out of the ribosome, but the capacity of the trigger factor is only like 100 amino acid or 150 amino acid. So if the longer amino acid chain is emerged out of the ribosome, then the trigger factor dissociates from the ribosome, but still keeps the client protein. So in that case, the trigger factor in solution, trigger factor in the absence of the ribosome can exist, and it can be the functional correlation with the other molecular chaperones, including Seg-B. So my answer is that even the trigger factor can work post-translationally.
There's one more question from Eloidam. Is there any disulfide bond in the TF dimer? If so, how many are there?
Thank you for the question. There's no disulfide bond in the trigger factor. So there's no cysteine residue in the in trigger factor.
Okay, thank you very much, Dr. Saio-san. And let's move on to the next speaker.
Thank you very much.
Our next speaker is Dr. Takaomi Saido. Dr. Saido completed bachelor's degree in biology from Tsukuba University and a master's and PhD in pharmaceutical sciences from the University of Tokyo. From 1988 to 1997, he was a research scientist at Tokyo Metropolitan Institute of Medical Science, and he became the laboratory head at Riken Brain Science Institute, Wako Saitama in Japan. He was a visiting professor at many institutes, including the Scripps Institute in US. He has received many honors, including the Outstanding Contributor Award from Alzheimer's Research Forum in 2004 and Toshihiko Tokizana Memorial Award in 2007. His research mainly focuses on protein misfolding and pathology of neurodegenerative diseases. Thank you very much for joining us. The floor is yours, Dr. Saido-san.
Thanks for the introduction. The title of my talk is GPCR-based treatment of preclinical AD. Why preclinical AD? AD is a disorder to prevent rather than to cure because neurons cannot regenerate after degeneration. And I'd like to talk about significance and of success of lecanemab endorses the A-beta hypothesis in clinical terms as a proof of concept, in addition to pathological, genetic, and experimental evidence. I thus joined the scientific community's appeal to FDA for early approval. A-beta protofibrils, rather than monomers, appear to be the major culprits because the antibody has about 1,000 times greater affinity for the former than for the latter. There are some issues to be addressed. Antibody administration makes A-beta deposition fully disappear, but the improvement of cognitive impairment is limited, about 27%. Is this because neurodegeneration and other pathological processes had already started? Or there may be an alternative hypothesis, as will be shown in the next slide. Anti-A-beta treatment will be most effective if used on preclinical AD patients. The number of preclinical AD patients in Japan alone is approximately 20 million. So the socioeconomic costs will be high, about $600 billion in Japan alone. And lecanemab causes such side effects as edema and hemorrhage. Lecanemab maybe resembles Wright brothers' airplane in that it is an invention that will yield more effective, specific, safe, and economical anti-A-beta medication in the future. So here's my hypothesis. When you have A-beta plaques in the brain and you expose it to a PET ligand, in this case PIB, you get binding of the PET ligand and you get positron emission. But when you have antibodies bound to the plaque, then PET ligand cannot bind to the plaque. There is no signal. So this is my working hypothesis as to how anti-A-beta immunotherapy works, but I don't have any direct proof. But it would be very easy to examine this hypothesis if I had antibodies. So there is some biochemical basis. One is extraction of A-beta from human brains requires high concentration of formic acid, the most acidic organic acid whose acidity is comparable to that of hydrogen chloride. Human plaques, which means human plaques are as hard as rocks. There are some cellular bases. The binding of the antibody can still interfere with the interaction between A-beta protofield and alveolus and glial cells. As a consequence, lecanemab administration reduces GFAP levels in plasma, which means the interaction between A-beta and astrocytes was blocked. So let me first introduce the overexpression artifacts in teenage mice. There are intrinsic pitfalls of APP and APP-PS1 overexpression paradigms in modeling Alzheimer's disease. That is overexpression artifact. There are two uncertainties of overexpression paradigm causing problems in preclinical studies. One is the uncertainty regarding why the artifacts arise. Second, the uncertainty regarding how the artifacts affect interpretation of experimental results. So we generated the very first APP knock-in mice in which the A-beta sequence of mRNA A-beta is humanized, and we introduced Swedish mutations that increases the total amount of A-beta, and Iberian bioreactor Iberian mutation that increases the ratio of A-beta 40 to 40, where A-beta 42 is the primary pathogenic agent. And then this results in overproduction of A-beta 1 to 42. And we also generated knock-in mice carrying the Swedish mutation only as a negative control, because this line does not accumulate A-beta. So we call this line NLF mouse line. And as you see, that the mice start accumulating A-beta at 9 months. And you see very intensive deposition at 19 months. But for grad students and postdocs, 18 months are too long to wait. So And this is how the three-dimensional image looks like, where the green represents the plaques and the red represents the red blood vessels. So we generated another second generation looking mice in which Arctic mutation was introduced. This makes the A-beta oligomerization prone. And we call this mutant line, NLGF line. And the mice start accumulating A-beta at 2 months, so about 3 times faster than NLF mice. And in addition, the Arctic A-beta is more pro-inflammatory than wild-type A-beta, as you can see here. And now our mouse models are used by more than 600 academic groups in the world. The continent where the mice is not used is African continent, where people do not live as long as in the industrialized continents. But I think African scientists will soon be using mouse models because their populations are aging. So our mice, using our mice, more than 200 papers were published, including these important papers. And here's an example of overexpression artifact. When APP-TG mice overexpressing APP was crossbred with knockout mice deficient in calpastatin, which is an inhibitor of calpain, calcium-activated protease. And we compared the phenotypes APP knocking mice with without calpastatin. And then we found that APP-TG mice crossbred with calpastatin deficient mice die very early. Half of them died in 10 weeks. But APP knocking mice that show as much pathology live as long as wild-type mice, even in the absence of calpastatin. So calpastatin deficiency induced early lethality in APP-TG mice is an artifact. And so there are more and more mouse lines to share, as you see here, and also some other lines. And I like to talk about the mechanistic link between A-beta amyloidosis and tau pathology. There are a number of mutations that cause tau pathology in exons and introns of tau gene. And we have generated these different lines of single and double mutants. And these mice show tau pathology in an aging-dependent manner. PS19 is a kind of positive control mouse line that overexpresses mutant APP, mutant tau. And now we have a new tool to resolve A-beta tau axis. This shows mutant wild-type tau knocking mice, and this is mouse that accumulates A-beta, but where tau is in the wild-type status. This is mutant tau knocking mouse that carries P30, P301S mutation and intro10 3 mutation, and they do not really show any pathological features. But when you cross that mutant tau knocking mice with APP knocking mice, you see a massive A-beta tau aggregation. So, and this is age dependent, as you see here. So what we can do is we can perform single-cell RNA-seq identification of cells and messenger RNAs, proteins, differentially expressed by subtraction, identification of the target molecules by introduction of shRNAs using AAV9, establishment of cause and effect relationship, and elucidation of mechanisms. So we have made more efforts to make better models, non-human private models, for which we use common marmosets, and common marmosets are closer to humans than mice, and they accumulate A-beta much earlier than humans after the age of 7. And we introduced the tau mutation, PS1 mutation by exon skipping. There's a 3' splice site here, and we use the TALEN for genome editing and destroyed the 3' splice site and induced exon 9 skipping, as shown here in RT-PCR. And PS1 autoclaves 8 cells into 27 and 70 kilodalton fragments. But when exon 9 is absent, this autoproteolysis is stopped. And as you can see here in that heterozygous mutant mice marmosets, you would see a full-length PS1 fragments. And as a consequence, the ratio of A-beta 42 to 40 significantly increased in the fibroblasts taken from the marmosets, which means the marmosets will develop early Alzheimer's disease within a few years. So we will make them available to those whom we can trust in the future. And finally, the main topic for today. So we know that the familial AD is caused by upregulation of anabolism A-beta. We do not know what is causing sporadic AD, and we hypothesize downregulation of catabolism is maybe a cause. So we analyzed A-beta degradation by injecting internally radiolabeled A-beta peptides into mouse live mouse on the rat hippocampus and analyze the degradation by radio-HPLC. And as you can see here, the A-beta 1 to 42 was degraded in a time-dependent manner. And using a panel of protease inhibitors, we identified neprilysin as a major A-beta degrading enzyme. And neprilysin is a type 2 membrane-bound protein, and phosphorylation and dephosphorylation of serine-6 regulates activation of neprilysin. And indeed, when you introduce S6A mutation in NEP, activity is increased on the cell surface. And then last year, there was a GWAS study published indicating the presence of risk alleles in neprilysin gene. One is in the 5' promoter region that regulates the expression of neprilysin. And there is a missense variant, methionine H spalling, and this is present 2 amino acids away from the serine residue that undergoes phosphorylation dephosphorylation cycle. So we intend to examine the effects of this MHP mutation on the activity and the metabolism of A-beta. And this data shows that when you cross APP and LF with neprilysin inoculate mice, you see massive accumulation of A beta, indicating neprilysin is indeed a strong A beta degrading enzyme in vivo. And also, there are a number of papers that show that aging-dependent decreases neprilysin expression in human brain and in AD brain. So I think it is necessary to upregulate neprilysin activity in order to reduce the aging-associated A beta deposition. So we have showed in 2005 that the neuropeptide somatostatin, a GPCR agonist, activates neuronal neprilysin, but using activity staining. But it took us a long time to identify the mechanisms because there are 5 receptor subtypes, and 1 to 4 is expressed. 4 are expressed in hippocampus and cortex, whereas the receptor subtype 5 is expressed in pituitary. So this means there are possible combinations, 5 homodimers and 20 heterodimers. So we introduced deficient mice in SSTR subtypes, deficient in homodimers, heterodimers, and in this case, SST5R5 was excluded because it is selectively expressed in pituitary. And we found that no single knockout mice showed any change in the neprilysin activity, but double mutants deficient in SSTR1 and SSTR4 show reduced neprilysin activity, which means 2 of the 5 SSTR subtypes regulate neprilysin activity in a redundant manner. And consistently, when we crossed epipinokimas with SSTR and SST4 double knockout mice, we show increased A beta deposition. And also, we used the pharmacological mean by introducing CH275, which is a SSTR1 and SSTR4 selective agonist by osmotic pump. Then the CH275 increased NEP activity and removed A beta deposition in the APP-NLG mice. And also, somatostatin receptor heterodimer composed of subtypes 1 and 4 are exclusively expressed only in cortex and hippocampus. So I think we can expect that blood-brain barrier permeable agonists specific to somatostatin receptor heterodimer composed of subtypes 1 and 4 will prevent AD, and GPCRs are indeed good targets for medication. So what to do next is positive screening, then followed by negative screening and optimization of the compound structure, preclinical studies using the single APP Noki mouse model, which I showed in the previous slide, and subclinical studies using AD marmosets, which will accelerate the following clinical studies. And then we hope to obtain safe and economical AD medications. And we are looking for industrial collaborators on these matters. And these are collaborators, colleagues and collaborators inside and outside the laboratory, and these are the members of my laboratory. Thank you for your kind attention.
Thank you very much, Dr. Saido. Let's wait for a minute or so for the questions in Q&A. A great talk. So you mentioned the beginning about this lecanemab and recommending for FDA approval. You mentioned that it has 1,000 times greater affinity for product fibers than monomers.
Mm-hmm.
Any data on the oligomers of A beta?
There could be, but as much as this antibody was originally created by Lars at Uppsala University, and he discovered the active mutation that is a conversion of E to G amino acid in the middle of the A beta sequence, and the active A beta forms protofibrils, but I'm not really sure if active A beta forms a dimer or not, so I can't really answer to your question right now.
So is this antibody for all sorts of A beta isomers? A beta 40, 42, 39?
Yes. But more affinity for A beta oligomers. And it also binds to A beta aggregates, like you see here.
So in your annual, there's a question here. Okay, let's take a question from Q&A. There's a question from Zhao. Hey, does single mapped P301S KI mouse show any tau pathologies as it get aging? You can look at the Q&A.
Yes. So actually, this is P301S looking mice carrying another mutation in the intron, but somehow this mutation alone does not really cause any major aggregates. Somehow you need the presence of a beta in order to induce tau pathology. So I think there's some sort of mechanism in which a beta induces tau aggregation. But you need the mutation, because here, when there is a beta and wild type tau, there is no tau pathology. So you need both a beta pathology and tau mutations to reproduce tau aggregation. Yes.
Interesting. Any other questions? There's one question from Q&A from Valaidam. Great talk. Does lecanemab bind to FCGRs in immune cells?
No, I don't think so. No.